All right. Hello, my name is Javier Hernandez, and I am here to tell you guys about the gut microbiome project. So let's begin. Um, a microbiome is basically where um, bacteria lives, and a gut microbiome is the bacteria that lives in a gut. That's the domain. Um, Physiological conditions that's caused by the gut microbiome is type 2 diabetes and obesity. Type 2 diabetes, it's correlated with the gut microbiome due to increases in fiber intake, and that fiber intake causes insulin resistance due to an increase in number of the gut microbiota, because the fiber creates more um, bacteria. And in obesity is linked with the gut microbiome through which was tested with mice. And from the experiments shown, it shows that their body fat increased when they got more food, and they moved the biota from the mice into another mouse, and it made it fat, which gut microbiomes is correlated with increase in fats, as said by Davis 2017. Okay, so now we're going to talk about the dogs that we picked. We picked 2 German Shepherds, 2 Golden Retrievers. The reason why we picked them is so we could see if there's a difference in their gut microbiome due to breed. And we picked relatively similar ages in dogs, so we can rule out the age difference in them. All right. So for the week 1 that we ran the experiment, we isolated DNA using the QIAGEN kit from fecal matter. We followed the instructions of the kit as prescribed, and then we used the Zymo kit to isolate the DNA from proteins, lipids, and anything that could throw off the DNA count. And we also followed the instructions prescribed by the kit. So in week 2 of the experiment, we quantified and we also checked the concentration of the DNA. We checked the concentration of DNA using the nanodrop spectrophotometer. It checked for UV absorption and how concentrated the DNA was. So if we go over here to our results, only sample 1 had enough DNA to actually give us a proper reading. The concentration was 7.70 nanograms per microliter, and then at 260, the absorption was at 0.155. So the ratio afterwards was 2.42. The rest of the samples did not give us enough concentration. So afterwards, we continued with the kit and its instructions as prescribed. We used the Amplicon PCR to amplify the DNA. We did 25 cycles. All right, so as I said, we did 25 cycles. We did cycles at 95 degrees Celsius for 30 seconds in order to denature the DNA, and then we did another one at 55 degrees Celsius for 30 seconds as well in order to get the primer in there. And then we did another one at 72 degrees in order to produce DNA synthesis. All right, so in week 3 of the experiment, we took the DNA that we produced in week 2 and put it into the gel electrophoresis. Over here I have a picture that shows our gel sample. Over here you can see the wells, the cathode and the anode, and you can see that we did get some DNA, but it was not enough. So, continuing with that, we got all the DNA that we had, and then we put it all together, and then we used a PCR index in order to stop the production of more DNA. Okay. All right. So in week 4 of the experiment, we performed the same steps as in week 3, but we removed magnesium ions primers in order to use a different tool to get a more accurate reading of the DNA. We used a tool called Illumina Mizik, which was a next generation DNA analyzer. And if we did not remove the magnesium ions, the primers, the NTP, then we would get wrong readings because they are considered impurities and can contaminate the DNA sample. So we'll go back to week 3. We did 8 cycles, and our temperatures for the cycles were 95 degrees Celsius for 30 seconds, 55 degrees Celsius for 30 seconds, 72 for 30 seconds, and then we held it at 72 degrees Celsius for 5 minutes, and it was completed at and held at 4 degrees Celsius. We did the PCR index. Right. So we do the indexing in week 3, and that is relative to the amplification of DNA of week 2, because the indexing stops the reactions and it gives us what we're looking for, which is the amplification of DNA. So in week 4 we use a fluorimetric assays instead of using a spectrophotometer, the UV from the spectrophotometer, because the UV spectrophotometer does not provide us with precise enough data. Like, it could be due to dilutions and certain factors such as those, whereas a fluorimetric assay does not do that. It's actually based on quantification, which is more sensitive and is often specific on the nucleic acid of what you're interested in. So our results on the Applicon PCR reactions was that we didn't have enough DNA from the very beginning, so our results were actually nullified. And then for the other part, we again, it was nullified, so we weren't actually able to have proper results. When it came to these parts, we actually used the results that were given from the last semester of the last year. Alright, so here are our results for kingdom, order, and genus. We had 10,889 proper reads. I just want to state that these reads were not ours. They were done by another student last semester or last year. And so light blue is for the young German Shepherd. Orange is for the 3-year-old Golden Retriever. The gray is for the 23-month-old Retriever. The yellow is for the German Shepherd, the older German Shepherd, and the dark blue is our positive control. And this was for the kingdom. All of them fell in the same kingdom, and in our order results, it's when we started seeing variances, but most of that was due to the positive control. And then in genus, our results, they started varying a little bit more due to the different breeds that they were. It's not on here, but there's another category which is other, and that's like other and unknown. That's for things that these kits were not able to pick up. So if it's a bacteria that's not known for that kit, it won't pick it up and it'll just put it under other. So I want to talk about our DNA extraction. It was not successful in the terms of after using the Zymo kit, because we only had one sample, which is sample 365, that was able to produce enough concentration. In order to be considered to have enough concentration, you have to be at least at 6.7 nanograms over microliters. And experiment 365 had 7.7 nanograms over microliters. And then we move on to our UV. The recommended was 1.5 through 2.2 for the ratio. And as per terms of this, our samples were actually had enough. The ratio was actually just right for our DNA. All right, so back to our gel electrophoresis. Our group is on the left, so the only one that shows is the positive control. The positive control was the only one that had enough DNA to show a band. The absence of a band at 550 is like we just didn't have enough DNA, and this happened to the entire class, for the most part. So no DNA was shown. So now we're going to the results of the index and PCR cleanup. We only had enough for the positive control, which was at 38.2 nanograms over microliters. In order to have enough, you're supposed to be at 6.7 nanograms over microliters. And for the rest of them, we did not. It was actually too low to read. Alright, so we expected to get nothing from the negative control, and from our results, we got nothing. Alright, so for our positive control, we actually had 2 successful reads, but and the results that we used from the last semester students, they actually got 10,889 positive reads, and 100 of them were successfully read. And with that being said, it meant that it was reliable to be used on the computers. All right, and so from the samples that we received from the last semester, which included the positive control, showed us that in the kingdom level, bacteria was consistent. It was very consistent, and they were all bacterium. And all the samples had adequate numbers and percentages. They all had over 1,000 reads, and they were at 100% bred. And none of the percentages were low or anything like that. If viruses would have been in the mix, then our readings would be thrown off, and that would be an indicator of cross-contamination. So at the end, if we come down here, at the end of our discussion, our initial question was, will there be a difference in breed of the dogs have an impact/effect on the gut microbiome? Thus, my group hypothesized that there will be a correlation difference in the diversity of the gut microbiome based on the different types of dogs that we have. The data collected for these samples do not support the hypothesis, because from the results it can be concluded that there is a difference in diversity of the gut microbiome, but breed is not a factor that determines determines the difference. Furthermore, we reject our hypothesis because of the data we collected in this experiment. Okay, and this concludes my presentation. Thanks for watching.